Oh, thank you very much. Now, I wonder, is it possible to bring up the lights for a moment? Excellent. I just want to find out who's here. How many of you are students? All right. And fantastic. How many are medical students? And those of you who aren't medical students, are you biological students and some other sciences? Let's see the biological students. All right. Fantastic. Look, you can leave them up for a moment. It's okay. More than anything, what I'd like to do is express to all of you who are students something that you can't quite know yet. When you just come into a field, the one thing you can't know is just how rapidly things can change over the course of time. Because you're still at the point of like learning all the things you're supposed to learn about medicine or learning all the things you're supposed to learn about biology. And what's amazing is just how rapidly fields can move. You drop a pebble into the water, and the ripples are small at first, and then the ripples go out and out and out and out. And over the course of a career, how big those ripples can become. So what I want to do is tell a story today. I really want to tell the story of genomic medicine and how sets of ideas that a group of people, my generation had, starting in the mid-1980s, have played out over time. And things that I've got to say we never imagined would be possible have gone far beyond what we ever thought could happen. And the reason to tell this story is partly because they're interesting facts and all that, but mostly because the same thing is going to be true for all of what you are doing. And that's what I really want you to take away. And so that's why I was so excited to come to India, because the advertisement was that I would get to talk to lots of students. And so this is for you. All right, let's start. And you can leave some of the lights up if you want to, as you choose. You'll let me know if we can see things. Excellent. All right. So I've entitled this, The Genetic Basis of Human Disease, a 35-year journey of genomic medicine. And what I'm going to do is touch on essentially 4 chapters of biological history. How it is that we went from having no idea about how to find the genetic basis of disease to being able to do it in a much more rapid and routine fashion than we ever thought possible. I'm going to start with chapter 1, the birth of genomics. The idea that arose in the early 1980s that there might be systematic ways to find the basis of disease. Now, for this you have to remember that back in the ancient world, like the 1970s or earlier, it was the case that if you wanted to find the basis of a disease, you just had to be like really smart or really lucky, because there was no recipe that would let you do it. You might be inspired to find some metabolite in a patient and trace down that the cause of the disease was some problem with that metabolic circuitry or something, but there was no particular way you could know how to do that. And yet, the idea became that you might be able to scan the entire human genome and find the gene that was responsible or contributed to a disease. Now, at first, this seems a little crazy. The human genome, 23 chromosome pairs, 3 billion letters of DNA. A typical gene is 3,000 letters. A typical mutation in a gene is 1 letter. It'd be like finding a needle in a haystack. Actually, if you do the calculation, it's worse than a needle in a haystack. 1 needle is actually more than 1/3 billionth of a haystack, and so it's actually, it's worse. So how are you going to do this? Well, David Botstein, a very, very smart biologist, actually a yeast geneticist, had a very simple insight around 1980, published this in 1980, that if you had genetic spelling differences between people, and you had a family in which a disease was transmitted, you could trace a genetic spelling difference and see if it co-transmitted in the family along with the gene responsible for a disease. That is, if a spelling difference showed the same inheritance pattern as the disease. If it did, you would know that the spelling difference had to lie on the same chromosome as the disease, not just on the same chromosome, relatively nearby on the same chromosome. And that in that way, you could pin down the gene to a specific spot in the genome. So I remember the day that I walked into David's office, and he shuts the door and he says in a hushed whisper, "The gene for cystic fibrosis is on chromosome number 7." Because somebody had found a linked genetic marker that co-transmitted along with cystic fibrosis. Now, it wasn't a perfect co-transmission, but 85% of the time, they would travel together in meiosis in a family. Within a few months, people had narrowed it down and found a marker that showed 99% correlated inheritance with cystic fibrosis. That's pretty good. Only 1% of the time would they recombine. You must be pretty close. The problem was 1% still translated into 1 million letters of DNA. And 1 million bases of DNA was a huge distance to cover back in those benighted days of the 1980s. You could only move forward at steps of about 10,000 bases at a time. And in fact, people worked on it, and it took 4 or 5 years, and it took maybe 100 people working on this, and it took many tens of millions of dollars. But eventually, people got from a nearby genetic marker to the gene for cystic fibrosis. And when they got there, this is what they saw. They saw a sequence, a whole lot of letters scattered around like this. But I call your attention in particular to those 3 letters, CTT over there, that encodes a phenylalanine at position 508 of that particular protein. And that's missing in the vast majority of patients with cystic fibrosis, and that's the cause of the disease. And one could do a genetic test to identify carriers of that mutation. Not just that, you could ask, what does this protein do? By tossing the sequence into a computer and asking the computer, ever seen any proteins that resemble this sequence? And the computer comes back and says, oh yes, this looks like many other proteins that sit in a membrane and transport things. Congratulations, you've probably just discovered a transporter, and that's probably the basis of cystic fibrosis. Of course, you have to go prove it. It's just a hypothesis, but it's very nice to be handed a hypothesis on a silver platter that you can then go test. And in fact, it worked out. So this was very good. And then as you began to think about it, and this is where I became involved, I met David Botstein around this time, you realized it could be applied not just to mapping simple single gene diseases in a family and with multiple affected children, but we realized it could also be applied in cases where families were consanguineous, where there was inbred marriage. And you could look for individuals who had homozygous particular mutations, and you could do that without even needing the family. The individual alone would be enough for that. And then it also occurred to us that you might be able to do this not just in a family, but in a population, like, say, Finland. Finland turns out to be a population isolate that was founded about 2,000 years ago, and almost anybody who has a particular genetic disease in Finland is carrying a mutation that entered into Finland 2,000 years ago, and all of the affected people have the same mutation, not all, but almost all. And that you could look for that too, and there'd be a smoking gun signature of that. And you'd need more and more genetic variants to recognize those signatures, but we came up with a bunch of ways to figure out how you could find the genes for simple one-gene diseases. And then it occurred to us that, well, you could do better than simple one-gene diseases. You could also take on polygenic diseases, diseases in which multiple genes played roles, and that you could treat an entire huge population, not just an isolated population like Finland, but all of Europe or perhaps all of India, as a population in which you could say, do the people who have diabetes tend to have an excess of one particular spelling of a gene relative to the people who don't have diabetes, and look for that association across a whole population. Around this same time, others were thinking about the applications to cancer. So there were some influential articles written in the 1980s that said, you know, we could also find the genes that drive cancer if we could compare the DNA sequence of tumor cells to the DNA sequence of the normal cells in the same individual. So as early as the mid-1980s, it was very clear that in principle, using genetic variation, you could map the genes for single gene traits, multi-gene traits, find genes underlying cancer. There was only one problem with all of this. The human genome is very, very big. And it just wasn't practical. The example I told you of cystic fibrosis was just one disease, and it took, zillions of people and zillions of dollars just to get one simple disease. If what you had to do was now look at thousands of diseases, this was just crazy. You couldn't do it. And so it became clear that what we really needed, chapter 2, was a human genome project. To be able to lay out all of that information and use it to be able to map disease genes. So a human genome project would involve, and this is what the scientific community debated in the 1980s, maps of genetic spelling differences up and down the chromosomes at pretty high density. Then all the underlying pieces of DNA, so you wouldn't have to do this slow process of moving along the chromosome as it had to be done with cystic fibrosis. And then actually all the sequence of all the bases. And not just that it would be marked up and annotated with where all the genes were. And not just that, but that all of that information would be freely, totally, immediately available to people anywhere in the world, whether in the United States or India, whether in academia or in industry. That was the plan. And the scientific community came together and said, let's do it. Well, it took changing the way we did biology, because if you did a little calculation in the mid-1980s, the amount of time needed to complete this project, based on the rate at which things had been going, was about 200 years. 200 years, and I think the young people in the audience will agree, is a very long time when you're thinking about your career. It's actually long compared to the kind of grants you can get and things like that. So it meant we would really have to come up with some ways to dramatically accelerate this so it didn't require 2 centuries. So genome centers were built. I had the honor of directing the largest of those genome centers under the Human Genome Project, where we thought about how to accelerate this, to automate this, to do new chemistries. And this is a picture of the production floor. It looks sort of like a Henry Ford factory. And we had these plastic plates, going around on conveyor belts, and we were so proud we did 250,000 pieces of DNA per day. We were doing this in Boston. At the same time, there was efforts not just in the United States, but in the United Kingdom and Germany, France, Japan, and China came in at the very last bit of the project and did about 1% of the project, which was a really important thing for them in terms of launching China in the world of genomics. So all of these countries came together, and we all agreed to the same principle of making the data freely, totally available, and it was a good thing. And the Human Genome Project got done. It got completed. We celebrated. In fact, it was so much fun. We celebrated multiple times over multiple completions. The Human Genome Project was finished in June of 2000 because the US President and the British Prime Minister said so. They held celebrations at the end of June announcing the completion of the Human Genome Project. In truth, for those of us who still like scientific niceties, like there wasn't any scientific paper supporting this claim. There was a lot of data on the web, but you really couldn't support the claim from what was there. But anyway, there was a big announcement about that. Then, about 5-6 months later, February of 2001, a paper was released. We wrote a paper reporting the sequence of the human genome and scientific analysis of that, and we celebrated that too. But of course, that was just a rough draft sequence of the human genome. It still had only 90% of the sequence, 10% was missing. It had 250,000 gaps. It had lots of errors. It was a really good thing, but it was a scruffy draft of the human genome. And so the International Consortium went on to make a finished sequence of the human genome. Footnote, finished is a technical term. It doesn't mean finished. Finished means we've done all the bits that can be done with current technology, but I feel obliged, in truth in advertising, to say that there are still little bits of the human genome we can't sequence. It represents a small fraction of it, but anyway, there are still 300 gaps left in the genome. And we celebrated that. In fact, we worked very hard to complete the finishing of the human genome by April 25th, 2003. Why April 25th, 2003? Because that was going to be the 50th anniversary of the publication of the Watson-Crick paper on the double helix. And we thought it would be so cool to get it done and throw a party on the 50th anniversary, and we did. And by the way, Watson and Crick were both still alive. Jim is actually still alive today. Francis has passed on, but they were both alive, and they both said at the time that they never in their wildest dreams did they imagine that they would live to see a full sequence of the human genome. That's what took place over the course of that lifetime. So, all right, so that's the scientific foundation. Now, before I leave chapter 2, I want to talk for a moment about the social and political and public policy issues that arose during this chapter 2, because they're important too, and as scientists, we ought to pay attention to those things. So I've described the project as this international effort. Well, it's also the case that there was a private effort to sequence the human genome by a company called Celera Genomics that was organized in the spring of 1998 after the project was quite far along. And they said, we're going to come along and we're going to do the sequence of the whole human genome under a different model. The model would be that we'll use private funds and it will be a subscription service and you'll pay money to access it. We didn't like this model. And this was an interesting competition that then arose over the next several years between the idea of a free public good versus a subscription model. But they're both plausible models about how you might choose to organize science. And when we published the paper in February of 2001, we had coordinated it that the Celera group published their paper at the same time. They were in different journals, Science and Nature. Originally, they were both going to be in Science. And I asked the editor of Science, are you going to require that they make the data available? Isn't that the deal on publication, that data become available? And Science said, well, yes, that's usually the deal, but this is so important that we're going to go along with their restriction that the data are not made publicly available. And we threw a fit about this and said, that's not okay. And I then called up the editor of Nature and said, would you be interested in a paper on the sequence of the human genome? And they said yes. And so we actually published that paper in Nature rather than Science because there was this question of, is it a public good or a private good? There was also a question about human genes and should they be patented and owned? And in fact, here, there were multiple efforts to patent human genes. The National Institutes of Health itself filed patents on little snippets of human genes, starting in the early 1990s. And most famously, when the genes for early onset breast cancer, BRCA1 and BRCA2, were mapped and cloned, patents were filed on those and licensed exclusively to a company in the United States, Myriad Genetics, that would then provide testing for women who wanted to know if they were at risk for early onset breast cancer. The issues with this test, of course, were it cost $3,000 per person to be tested, and therefore was not accessible to the vast majority of people. And then another issue to be thought of in terms of science is just plain hype. The New York Times, with a screaming headline across the entire front page on June 27, 2000, announced, "Genetic code of human life is cracked by scientists." You know, breathless promises about what was going to happen because of this. One could plausibly imagine that all disease was going to be cured in the next couple of years because of these amazing discoveries and all that. And Time magazine loves to put this stuff on the covers. So that's the end of chapter 2. Now the problem was at the end of chapter 2, we couldn't possibly live up to all of this hype about what it was going to do because what we had was 3 billion letters of DNA in front of us, and we didn't know how to read it. You know, it'd be like me getting a book written in Tamil. I might have all the letters, but it's not going to help so much because I don't read Tamil. So chapter 3, the human genome coming of age. Really the work of the past decade has been the scientific community coming together to interpret this human genome. To take pretty boring sequence, A's, T's, C's, and G's, and shine spotlights on particular parts of the genome and say, aha, that encodes a portion of a protein, and that's a regulatory sequence, and other things, to breathe meaning into this meaningless text. For that, the first thing you want when you have languages you don't speak is a Rosetta Stone, comparisons between languages. And so for us, the Rosetta Stone was the mouse. Now, we didn't speak mouse either, but it turns out that you can, even though you don't speak human and you don't speak mouse, you can look for bits that are very similar between human and mouse and say, if something is biologically very important, the odds are pretty good that evolution has preserved it over the 100 million years since our common ancestor with mouse. And so by lining up these two genomes, you can ask, where are the evolutionarily conserved bits? And we got a lot out of that. So much so that after we compared the human and the mouse, and we had a sequence of the mouse pretty soon thereafter, we got the rat and the dog. And we were learning so much from the human and the mouse and the rat and the dog that we got us, you know, the elephant and the porpoise and the cat and the this and the that. And we ended up getting 48 different vertebrate species and lining them up and trying to read this whole signal of evolution about what was really important in there. What did we learn from all this? The best way to illustrate what we learned from this is to tell you what we knew in 2001. Now, I know what we knew in 2001 because this has been mentioned. I teach freshman biology at MIT. So I have notes from 2001, and I can say, what did I teach them, expect them to know on the test? Well, if you were taking the course in 2001, you'd be expected to know that the number of genes in the human genome was between about 35,000 and 100,000 or so, that typically the structure of a gene had the sequence of the protein as the primary part, and then some bits of regulatory sequence, but that was small compared to the structure of the protein. That almost all genes encoded an RNA that was translated into protein, with maybe a dozen or so weird exceptions of genes that were not translated into a protein. And also that the human genome was littered with transposable elements that had jumped around and were selfish junk DNA. Progress in science is that we now know, based on what I told you, that everything that I taught the freshmen there is wrong. None of those statements are correct. What is correct is that the number of protein-coding genes is much smaller, only about 21,000. We know that by comparison of mouse and human and rat and dog, which have shown that many of the things that we thought were protein-coding genes really aren't. We know that there are thousands of genes that are never translated into proteins. We know that there are millions of regulatory elements and that most of what evolution has lovingly conserved over the last 100 million years is actually regulatory sequence, not protein coding sequence. And we know that these transposable elements, these jumping genes, most of which are really junk, nonetheless, some of them play a critical role in carrying around the genome evolutionary innovations. All of this we learned by reading the sequence. Now, beyond that, technology. What has happened with technology is mind-blowing. The cost of sequencing, well, that first person that we sequenced in the Human Genome Project, we spent $3 billion on that sequence. That's a lot of money. Since then, though, the cost of sequencing a human being has plummeted to $3,000 and now below that point. It has fallen by 1 million-fold over the course of a little more than a decade. There is nothing in history that has ever become 1 million-fold more efficient over the course of a decade or so. Even the famous Moore's Law in microelectronics runs 3 times as slow as that. It takes 3 decades for Moore's Law to get 1 million-fold improvement. So with all of that, what's been happening is that first map of the human is now being complemented by zillions of other kinds of maps. Any technique you want, people are going off and making more maps and more maps and more maps. On top of the genetic and the physical and the sequence maps, we have maps of evolutionary conservation by comparing species. We have maps of chromatin state by looking at the modifications of the chromatin proteins in different tissues. We have maps of the 3-dimensional folding of the genome, maps of genetic variation, disease association, the regions that have been selected over the course of evolution during the human, maps of, just maps upon maps. It's like, you know, on your smartphone, having Google Maps, and on top of the map, you have the traffic, and you have the pizzerias, and you have other things, and they're all layered on top of each other. There's been tremendous effort to generate and make freely available all of those data. So how are we doing with respect to this question? The question that got me excited in the mid-1980s about could we find the genes responsible for disease? Well, let me give you a progress report. With respect to those rare single gene monogenic Mendelian disorders, when the Human Genome Project started in 1990, there were only 70 examples where we knew the gene underlying a genetic disease. Currently, it's gone up. 2001, it got up to about 1,300. By today, it's about 3,600 different genetic diseases where we know the genes that are responsible. There's still about an equal number to go, but it's moving at a tremendous rate. It's going very, very well. Moreover, there are some really cool examples. Now I'll get technical for a second for the medical students. Here's a nice example. In the Middle East, there's a tremendous amount of consanguinity, marriage between cousins. In southern India, there is as well, but it's been looked at more extensively here in the Middle East. We've collaborated with Joe Gleason, now at Rockefeller. And he's looked at many disorders, including this very interesting case of an autism associated with epilepsy. And what we found in this family, this is work of Stacy Gabriel at the Broad, was that they had mutations in a gene that encoded a kinase that put a phosphate on the enzyme that degraded branched chain amino acids. And when it put the phosphate on, it downregulated the enzyme. So hang on to your seats here. If you're missing the thing that downregulates the enzyme, then the enzyme is going to be too active, and it's going to degrade branched chain amino acids too much. So, hypothesis, if I fed a lot of branched chain amino acids to an individual, it might overcome this genetic defect. So they made the mouse model that contains that mutation, and sure enough, the mouse model, when fed an excess of branched chain amino acids, overcomes the defect that you see there, the neurological defect. And now they're trying this in humans. So this was a beautiful example where there may be a really simple dietary intervention that can have a serious effect on this genetic disease. Most are not so as simple as that, but it's a nice example. But now what about these complex diseases, the ones that are polygenic, multigenic diseases? How are we doing on those? Those are the ones that we all think about. Diabetes and schizophrenia and heart attack and inflammatory bowel disease. Well, they're very important because if we can understand the basis of these diseases, we stand a chance of trying to develop therapies. So the purpose of finding the genes for these diseases are to get targets for therapeutic development, and not just targets for therapeutic development, but actually to be able to test therapeutic hypotheses. And I'll give you some examples for the medical students about what I mean by testing therapeutic hypotheses. But briefly, what I mean is this. Instead of spending $1 billion to develop a drug and then run a clinical trial and find out, was this drug a good idea to have developed, wouldn't it be good if you could see in advance what would happen if I inhibited a protein? Would it have a good effect on a disease? So for example, could I find individuals in which the protein of interest, the target for a presumptive drug, was knocked out, and ask were they protected from the disease? In a way, I'd be running, nature would have already run the clinical trial for us even before we developed a drug. That would be a pretty good thing. So keep that in mind, because let me tell you about some of the cases that have emerged here. Well, I'm going to skip over the fact that it took about 6 years of work for the scientific community to come together and create all the tools, maps, super dense maps with millions of genetic variations, et cetera, et cetera. I'll take that for granted, but it was a lot of work. What it's led to is the discovery of more than 4,000 genes associated with more than 300 different common traits and diseases, and I'll show you some examples. Age-related macular degeneration. In a tiny sample, this is not typical, it was a very lucky case, but in age-related macular degeneration, a sample of just 150 people revealed that this disease is really about the alternative complement system. that 3 genes, complement factor H, complement factor B, and complement component 2, were all shown to have genetic variations that were associated with age-related macular degeneration, and it's completely changed the way that people are now thinking about developing therapies for age-related macular degeneration, is they're now developing molecules to tweak the alternative complement system. 150 people, wow. That was so totally misleading because almost no disease since then has been so easy. Here's a really typical case. Well, yeah, maybe it's a particularly hard case, but anyway, schizophrenia. At the Broad Institute, some of my colleagues, Mark Daly and Steve McCarroll, did a study of 3,000 cases and 3,000 controls and did in each person 1 million genotypes. And they found absolutely nothing. Not a single thing significant. And that's pretty depressing. Any of you students who've been involved in medical research, that'd be pretty depressing. And at the National Institutes of Health, when the study sections say, oh, should we support work on this? And you do this big study and nothing comes out of it, they say, well, we're not going to throw good money after bad. But you know, they looked closely at the data. And what they saw in the data was kind of curious. The p-values, the significance levels for each of the different genetic variants, if there was nothing there to be found, they would be distributed uniformly between 0 and 1. But they weren't. They were piling up. There was an excess of p-values that weren't yet significant, but were trying to be significant, piling up toward significance. And what a sophisticated person would say there is, even though it's not significant yet, I predict if I make my sample size larger, I'm going to be able to see things. So they expanded the sample size. Thank goodness we had philanthropic support from a very forward-thinking philanthropist, because the NIH would never have funded this. But they took it to 20,000 and found 5 significant loci. And then they took it to 50,000 and found 62, and now to 110,000 people analyzed. And there's 108 significant loci involved in schizophrenia that have now been found. It's good they kept going. And they include genes encoding voltage-gated calcium channels and genes related to glutamate signaling and sensible and interesting pathways, most of which were not previously recognized. And now, in addition to these studies, which I've described, which have to do with common genetic variations, they've now been doing studies of rare genetic variations and finding similar genes coming up. Similar stories with regard to heart disease and lipids. Here, beautiful work of my colleague Sekar Kathrison, where they've found more than 160 genes related to LDL cholesterol and HDL cholesterol and triglycerides. And about 60 genes related to early onset heart attack, because we know that many people die early of a heart attack, and for, at least in the United States, for 50% of people who die of a heart attack, the first presenting symptom is death. There are no prior risk factors that are known other than family history, and that's a very bad situation. So anyway, what do you do with all these genetic variants? Now, let me digress for a second, because I have a whole bunch of medical students. Medical students, you all must know that LDL is bad and HDL is good, right? Yeah? Okay. That's an epidemiological observation in the population, that people who have high LDL have much higher rates of heart attack. People who have high HDL have protection against heart attack. So, as you well know, people develop the statin drugs to lower LDL, and it decreased the risk of heart attack. That's good. And then a lot of drug companies said, great, let me find drugs to increase HDL, and that'll also protect against heart attack. The problem is correlation is not causality. Just because HDL happens to be high in people, or in people who HDL is high, they have protection against heart attack, doesn't mean it's caused by HDL. Maybe there's something else which is protective against heart attack and also raises HDL, but raising HDL won't do the job. You think I'm being churlish here, but no. So can you test that? Yes. Using the genetic variations that people inherit that increase their HDL, you can take a set of people and ask, do the people who inherit Genetic variations that raise their HDL show protection against heart attack. Answer, not at all. For LDL, you see that. For HDL, you don't. Therefore, it's probably not a good idea to develop drugs to raise HDL because they're not going to work. That would have been a really good thing to say to the 4 drug companies that each spent more than $1 billion developing drugs against HDL, the first 2 of which have already failed. So genetics can be useful. Now, what's really nice, if you want better targets, is these various genes here, what's been found is mutations that destroy the gene and protect against heart attack. People who have null alleles in PCSK9, ApoC3, or NPC1L1 show lower risk of heart attack. That's already a clinical trial that decreasing the function of those genes will convey protection against heart attack, and companies are now working very hard to develop drugs that will mimic the effect of the human mutation. Similar things for type 2 diabetes. There's a beautiful work of my colleague David Altshuler showing that there's a loss of function mutation in a zinc transporter in the pancreas that confers 3-fold protection against type 2 diabetes. We're very relevant in India. There are the inflammatory bowel disease, rare protective mutations there as well. Now, there's a tremendous amount still to go. Most of what I've told you has not been full sequencing of genomes. It's been using the common genetic variants, but not yet deep enough sequencing to get all the rare genetic variants. And it's been done on relatively small numbers of people. And it's been done mostly in Europeans, very little work in South Asians or East Asians or Africans or Hispanics. So we've really only looked at a very limited slice of the population. But we need to start looking at this in much larger groups of people. And the only effective way to do this is going to be to learn in the course of clinical care, to really turn our healthcare systems into learning systems, so that all of you who are doctors or soon to be doctors should be thinking about how do we get all of that information in the course of clinical care, and we can learn from all of that. Because I think we're going to enter into a world where it's plausible to have genomic information on everybody, and it will be cheap enough to imagine doing such things even in an Indian context, because you only have to get it once for a person over their lifetime. You get to amortize it over the course of 60 or 70 or 80 years, and so when the cost falls to $600, it's $10 a year to have done such a thing. It's no longer crazy to imagine such things. We have to manage to collect all of that information and use it to drive discovery. Now, much more briefly, I'm going to say the same sorts of things have been happening with cancer. With cancer, we've got, the idea was a simple idea that you could look at tumors, look at the normal DNA, and ask which mutations arose in a cancer. And then you would ask which genes had a big excess of mutations, and they should be genes then that drive the cancer. Well, all that was a very clear idea, but of course it took high-powered sequencing to get there, so it's only been in the past 10 years that it's become possible to do those things. A consortium was put together, the International Cancer Genome Consortium internationally, and in the United States, the Cancer Genome Atlas, to take on the looking. And I gotta say, it was controversial, because there were many people who thought this would be a total waste of time. One of the good examples here, the Human Cancer Genome Project, one more misstep in the war on cancer. So it was controversial because I think many people said, look, we already know all the genes responsible for cancer. Why are we looking? That was about what half of the criticism was. The other half of the criticism was, cancer is so hopelessly complicated, we're never going to know all the genes. Why are we bother looking? It would be great if you could get the two of them to argue with each other first, and so you'd only have to face one of those. But so it's basically we already know everything, or we're never going to know everything. And we took the sort of moderate position of we probably don't already know everything, and why don't we have a look? And so eventually that was what happened. Was this Cancer Genome Atlas got off the ground, and to make a long story short, lots of new classes of cancer genes were found that people had totally missed before. Genes involved in such things as epigenomic processes, cell type specific transcription factors, genes involved in cancer metabolism, and we realized that an unbiased look that came through mutations could identify for us new types of cancer genes that we had missed. So now a fair question is, are we done yet? The short answer, no. The long answer is contained in this paper that appeared about a year ago from our group, where what we did was we looked at 5,000 tumors across 21 different cancer types. And we examined them closely for their mutations and asked which genes had mutations at a significant level. We looked for genes that had an excess of non-synonymous mutations or excess of clustering or excess of mutations in conserved patches. And just to show you, here's some 4 famous cancer genes, which you can't miss. The RAS gene, NRAS. This is the pattern of where mutations are. This is not a random pattern, right? This is not scattered about. And you can tell this is a highly significant cancer gene because the pattern is not random. Same for PIK3CA, highly non-random pattern. APC gene in colon cancer. Only pile up in colon cancer and only in this first half of the protein. And over here, retinoblastoma, lots more stop codons that destroy the protein. So by the patterns of mutations, you can learn things. And so to make a long story short, we looked at the patterns of mutations found in all the 20,000 genes in the genome, and we just say, are there more mutations than you'd expect by chance? Very brief digression. It's not so easy to say, what do you expect by chance? If you make a uniform background chance model, you get silly conclusions like, oh my goodness, in a small sample of lung cancers, there's 450 significant genes. But that's because your model is wrong. The mutations are not, the background rate is not uniform across the genome. If you correctly model the background mutation rate across the genome, then it all goes away and you're able to get it back down. So that's just a technical digression. But about 200 cancer genes emerged from this analysis, including 33 new cancer genes that had not previously been identified before in all sorts of very interesting and reasonable processes. So there are a lot more genes still to be found. Here's an example. This is a gene, don't worry about the details, but it's a gene that is like RAS, the famous cancer gene. It's a small GTPase, and 5 patients have exactly the same change in that protein at position 35, and that's a very important position of the protein called the effector domain of that protein. This couldn't happen by accident, highly significant. Here's another one of these small GTPase, and you've got mutations piling up exactly in the effector domain, and these had been missed, but you could see them. So what do we need? Well, again, we're only scratching the surface. We need much larger samples so we can fully squeeze out all of the information inherent in cancers. And increasingly, we're going to need to do this not just by collecting research samples, but doing this in the course of clinical care, turning our healthcare system into a learning system. And by the way, because you should think about doing this in India, we need to find not just the genes that are responsible for the tumor arising in the first place, we need to find the genes that are responsible for why a tumor that initially responds to drug later becomes drug resistant, by comparing the resistant tumor to the sensitive primary. And almost no such biopsies are done routinely. So we have shockingly little data. While 20,000 primary cancers have been analyzed, I think less than 200 resistant cancers have been analyzed. And that's just terrible. We need to get that sort of information. Briefly, I want to come back to the public policy questions I raised before and how they pertain to Chapter 3 here. First, this question, should this kind of information be a public good or a private good? The Human Genome Project approach or the Celera approach? Well, I think the situation is very clear now. Because all this information is publicly available, we've been able to make maps and maps and maps and maps, and people can layer things on top of those maps. And had each of these things been its own separate subscription database, it wouldn't be possible to mix and mash them together. So it's clear that from a scientific point of view, open data was the right thing to do. Actually, the National Academy of Sciences in the United States had a study where they went back and asked the question, should Science magazine have published a paper in which the data weren't made freely available? To their great credit, they concluded, no, that's just wrong, that shouldn't have happened. In fact, for those of you who are new to science and just coming into it, you probably are not aware that the whole idea of publishing papers in journals was a major innovation in the 1600s. Because before that, scientists kept their results secret because they had no way to establish priority, to say, I discovered this. So they would keep their results secret, and then they tried to somehow prove to you that they knew something by you could ask them a question and they could answer it, and that would prove that they kind of knew. What a crazy system. And so we developed publication. You get priority in return for revealing your data. And the idea of publishing a human genome sequence without revealing the data violates that. And the National Academy, to its great credit, said, yeah, that should not be allowed. We can't do that. It breaks the bargain of publication. With regard to patenting, should genes be patented in a way that could deny somebody access to finding at an affordable price information about their own DNA? I think all of us never expected to see this, but the United States Supreme Court remarkably concluded that genes may not be patented under US law. 9-0, a unanimous decision of the Supreme Court concluded that under US law, genes are products of nature, not inventions of humans. Really, it seems kind of obvious, but nobody was betting this way. And so it was really good. And then hype or hope. Well, the New York Times, with its banner headline, 10 years later, published a story saying, basically, human genome fails to cure all diseases. And what was really great was it was the same reporter who had done the first story who now did the second story. It's really great. But kind of more reasonably, I'm impressed by a study by the Battelle Institute, in which the Battelle Institute tried to ask what was the economic impact of these $3 billion that were spent on the Human Genome Project, and they roughly found that the economic impact was about $800 billion, something like more than 100-to-1 return on investment. Quite a remarkable return if you can get that over the course of a decade or so. But that's what science does. Science can pay returns like 100-to-1. And it's not hard to see where all of that comes from. I can point to individual companies that have emerged because of the Human Genome Project that alone have market capitalizations of $25 billion. So there are big impacts to it. So I think this notion that in the story, if Human Genome Project fails to cure all disease, well, actually, we never promised to cure all disease. Only the New York Times promised that we'd cure all disease. And so anyway, there you go. So what's Chapter 4? Chapter 4 is where we go from here. Precision medicine, and that's up to you. And that's why I'm so glad to be talking to so many medical students in one place, because you guys are responsible for this. This is your generation's work. We have to turn our healthcare system into a learning system. The National Academy of Sciences in the United States had a report that's worth looking at called Toward Precision Medicine. And the idea was that by collecting large amounts of information about DNA, but also information about microbiomes and information about gene expression and many different kinds, having it all in some knowledge commons, we'd begin to able to develop a new taxonomy for disease. A taxonomy for disease driven by the molecular features and dividing diseases up in ways that made them homogeneous as molecular diseases. In effect, it would be a medicine that would emerge from having access to this huge library of information. The library of information about the DNA of every species on the planet, the DNA of every individual within a species, the DNA from every cell within that individual, and not just the DNA, but the RNA and the protein. And you should imagine that every one of those cells has a book written about it. of its DNA and its RNA, and that increasingly we can have access to all that information. The question is, what do you do with that information? Some of what has to be done in the research laboratory. And there are amazingly cool tools that have come along for doing this in the research laboratory, ways to analyze single cells. A single student at the Broad Institute gave a talk where he explained that he's now able to read out the RNA transcriptome from 10,000 single cells per day. 2 years previously, you couldn't do any. And now he was giving a talk saying, I can do 10,000 cells a day. Ways of editing the genome with tools like CRISPR, very cool. Ways of reconstructing cellular circuitry, very cool. But these are things you can do in the laboratory. What we also need to do is really use the whole human population, to use the amazing variation in the human population. And for that, various countries have been organizing themselves. The United Kingdom has organized Genomics England and now Genomics Scotland that are assembling large samples of people who will be participants in a research study, 100,000 samples in the United Kingdom, with not just genome information, but lots of other medical records information. In the United States, a conversation started last year, in the spring of 2014, provoked by the president. The president himself became very interested in driving precision medicine, driving progress through making sure that we kind of had like an internet of information that could be used by physicians everywhere. And so he got us together to ask about what could the United States do in this, and it led just last month to the president announcing the launch of a precision medicine initiative. I like seeing the president of the United States there with DNA double helix, looks cool to me. And this plan is now going to roll out involving various kind of studies on cancer and creating a cohort of research participants, about 1 million research participants, and improving the way we regulate genetic diagnostics. And boy, we got a lot of challenges in the United States on this. Our medical records are not interoperable. They're very messy and complicated. We have issues around privacy. We have issues about whether we can recontact patients. It's going to be complicated. But the point is you take on complicated things because they force you to answer the questions. They force you to solve things. And I think that's why a precision medicine initiative is going on. But you don't have to be the United States of America or the United Kingdom to do this. Places like Estonia in the Baltics are launching an Estonian precision medicine initiative. They've got certain special advantages in Estonia. Their medical system has amazing medical records, and they launched a biobank with great foresight years ago. And in that biobank, which contains about 5% of the country's population, they can access and study electronically everybody's medical record. And when I was there, they apologized to me that the medical records are not completely up to date. They're usually about a week out of date. And I was just my eyes were rolling. A week out of date. Oh. So they have something special to contribute, and they intend to do that there. Now, we've got to make sure that the sharing of information here goes on in a way that involves the whole world. And so at the Broad Institute, we, together with colleagues, helped start something called the Global Alliance for Genomics and Health. It now involves more than 200 organizations in 28 different countries coming together to develop standards and to promote the sharing of genomic information. And we're working to launch a movement we call Count Me In, whereby patients will be able to check a consent that says, yes, count me in, I wish to share my genomic information. I want my privacy protected and all that, but I wish to share. Because I think, and many of you can ask as medical students what your experiences are, that most patients want something good to come of their experience. Even a cancer patient who's going to die, especially a cancer patient who will die of their cancer, wants something good to come of it, wants somebody to be able to benefit from it. And most such people want to be able to contribute to progress, and it is their right to do so. And we have to create ways where, if they wish to, they can do so. So that brings me to the last question, which is India's role. What are you all going to do about this? All you medical students and the doctors here. What should India's role be? You're not the United States, and you're not the United Kingdom, and you're not Estonia. Actually, you're about 1,000 times larger than Estonia. But you have some incredible distinctive things in India that nobody else has. You have a huge population with extraordinary diversity. When you think about the diversity across the Indian subcontinent, it's just amazing. You have endogamous groups that differ in all sorts of ways. You have wildly different environments and environmental exposures. If you had genomic information and medical records information, and you could correlate genetic variation against incidence of disease, against environmental exposures, there is so much that could be learned from India. India is potentially the most interesting country in the world in which to study these sort of genomic questions because of its size and diversity. There is already good work going on in India toward this, but there's not enough. It's not close to enough. You guys aren't yet doing what's really got to be done to truly meet this whole promise. And so one of the things I want to be doing, since I've got the next generation of Indian doctors here, is saying to you guys, think about how India should set some bold goals for what it can be doing here and how it will come back to benefit India. I leave you with that challenge because it's an important challenge. Just as with the Human Genome Project back in the mid-1980s, we had no concept that we'd ever get the whole sequence of the human genome and ever get to the point where we could read out all these genes and ever get to the point where things would be 1,000,000-fold cheaper. You medical students are saying, wow, this sounds kind of crazy, the idea that we could turn our healthcare system into a learning system and really get to the bottom of diseases and test treatments like this and all that. And you're absolutely right. It is crazy. Almost everything good starts with some crazy idea. And it turns out that over the course of time, when you drop that pebble in and it ripples out, those circles become very, very large. And so whatever you choose to do, whether it's this thing or other things that you're involved in, go pick bold and ambitious things to do. They're the only things worth devoting a career to. So putting up a final slide to acknowledge various people at the Broad who I've worked with and want to thank for some of the work I've described. I shall stop. Thank you all for your attention, and I will be delighted to take questions. Thank you.